Okay, section 12.8 deals with enzyme inhibition and regulation, and we're going to start out by looking at inhibition, and we're going to look at irreversible inhibition and reversible inhibition. All right, so an enzyme inhibitor is going to be any substance that can decrease the rate of an enzyme catalyzed reaction. And an understanding of enzyme inhibition is important for a couple of reasons. First, the characteristic function of many poisons and some medicines is to inhibit one or more enzymes and to decrease the rates of the reactions that they catalyze, of course. Second, some substances normally found in cells inhibit specific enzyme catalyzed reactions and thereby provide a means for internal regulation of cellular metabolism. Enzyme inhibitors are classified again as reversible or irreversible, depending on how they behave at the molecular level. Now, an irreversible inhibitor is going to be an inhibitor that forms a covalent bond with some kind of specific functional group of the enzyme and thus renders the enzyme inactive. So it just completely shuts it off. And a number of poisons, like deadly poisons, act as irreversible inhibitors. Things like snake venom, for example, or the cyanide ion is an irreversible enzyme inhibitor. It's extremely toxic and acts very rapidly. So this is why cyanide is so dangerous. And the cyanide ion, the way it works is that it binds to the iron cofactor of cytochrome oxidase, and it forms a stable complex with it and prevents it from functioning properly. And as a result, cell respiration stops and causes death in a matter of minutes. Any antidote for cyanide poisoning has to be administered quickly. One antidote is sodium thiosulfate. This substance, what it does is it converts the cyanide ion into an ion called a thiocyanate. And you don't have to memorize the structure of a thiocyanate. It's shown in the textbook. Um, but the thiocyanate does not bind to the iron of cytochrome oxidase. Another example that's shown here is that the toxicity of heavy metals like mercury and lead is due to their ability to render the protein part of enzymes ineffective. And what happens if you get poisoned with mercury, like you see down here, is that it can combine with the SH groups found in many enzymes and render them completely inactive. And again, you can see here that we're forming covalent bonds, and so those aren't going to let go. Now, not all enzyme inhibitors are going to act as poisons toward the body. In fact, some are used for therapeutic reasons, like sulfa drugs and the group of compounds known as penicillins are two well-known families of antibiotics, and I'm sure you've heard of them before, and they inhibit specific enzymes that are essential to the life processes of what? Not humans, but bacteria, right? So they kill bacteria. And the first penicillin was discovered way back in 1928 by Alexander Fleming when he noticed that the bacteria on a culture plate didn't grow in the vicinity of a mold that had contaminated the culture. And the mold was producing what? Well, it was producing penicillin. And you can see the structure shown here of ampicillin, which is the most common of the penicillins used. And the way penicillins work is that they interfere with what's called a transpeptidase, which is an enzyme that is important in the bacterial cell wall construction. And so, since the bacteria can't form cell walls, it prevents them from surviving, and so they die. A reversible inhibitor, in contrast to one that's irreversible, is one that's going to bind reversibly to the enzyme, right? So it's going to be able to bind to the enzyme. So there's our enzyme, there's our inhibitor, and we can get the enzyme inhibitor complex. But you see that this reaction is in equilibrium, so it can go right back to losing the inhibitor. And so the inhibitor can be removed by just shifting the equilibrium. And so there's different types of reversible inhibitors. There's competitive and non-competitive. And a competitive inhibitor is one that binds to the active site of the enzyme. And so it competes with substrate molecules for that active site. So again, it binds to the exact same site that a substrate would. So it's like a competition between the two. I guess I'm repeating myself. But anyhow, competitive inhibitors often have molecular structures, as you can imagine, that are really close to that of the substrate of the enzyme, right? They're kind of like mimicking the structure. And so the nature of the competitive inhibition is going to be represented in this figure that you see here. So there's a competition between the substrate and the inhibitor for the active site. And once the inhibitor binds with the enzyme, then the active site is blocked. All right. So we see here we have our inhibitor and somehow it's binding. And now you can see at first the binding site would fit the substrate, but now it doesn't fit the substrate. So it just shuts it off. Now, competitive inhibition can be reversed by increasing the concentration of the substrate. If enough substrate is added, it's eventually going to displace the inhibitor, and thus the competition between the inhibitor and the substrate is won by whichever molecular species is present in a greater concentration. Now, a non-competitive inhibitor is going to not look like the enzyme substrate whatsoever, and it's going to bind reversibly to the surface at an enzyme, but it's at a site that's other or a site that's not the catalytically active site. Sometimes we call these an allosteric site, and the interaction between the enzyme and the non-competitive inhibitor causes the three-dimensional shape of the enzyme and its active site to change. And so the enzyme is no longer able to bind to the normal substrate, or the substrate is improperly bound in a way that prevents any kind of catalysis and any kind of reaction. And you can see that from my previous slide that this figure was actually duplicated. So on the previous slide, there's actually a different figure that's shown in the textbook for a competitive inhibitor. I'll let you take a look at that on your own time, but that is figure 12.35. So what I should have had here is figure 12.35. So again, you can go back and take a look at that. But this figure 12.38 shows you a non-competitive inhibitor because it is an inhibitor, right? It's changing the shape of the active site, but it's not binding at the same spot. And so we say it's a non-competitive inhibitor. And like I told you, I don't know if this word is in the textbook or not, but when you have a non-competitive inhibitor binding at another site, sometimes we call that an allosteric site. Now, unlike competitive inhibition, non-competitive inhibition cannot be reversed by the addition of more substrate because additional substrate is not going to have any effect on the enzyme-bound inhibitor. That is, it can't displace the inhibitor because it can't bond to the site that's occupied by the inhibitor. And so we see the word here, allosteric regulation, or the definition, which is just when a substance binds to the enzyme at a location other than the active binding site. And then feedback inhibition is the process in which the end product of a sequence of enzyme catalyzed reactions inhibits an earlier step in the process. So it's like if you have a thermostat in your house and you turn the thermostat on and you set it to, let's say, 70 degrees. So when's it going to shut off? Okay, it's going to shut off when the thermostat reaches 70 degrees, right? And so that's kind of like a feedback inhibition, right? The furnace is going to run, it's going to keep heating and heating and heating until it gets to that level. And so you see what's happening here. We take this threonine, which gets acted upon by this enzyme and all these enzymes like this. Then you get to isoleucine. So once you get the amount of isoleucine that you need, what happens is that it's going to go back here and it's going to end up in some kind of binding site in this enzyme and it's going to shut it off. So it says here inhibits threonine deaminase. So it shuts off the mechanism. And so that's what we call feedback inhibition. Okay, and so a summary is always handy at the end of a video or a chapter, and there's a really good one here. So if we have some kind of enzyme inhibitor, so remember there's two types. There's going to be reversible and irreversible. So an irreversible one is going to be like a poison, like a cyanide. So we'll think here things like poisons are good examples of irreversible inhibitors. They bind covalently, they don't let go, shuts off the enzyme, and that's it. Then if we have a reversible inhibitor, right, we can it's going to be in equilibrium, right? We're going to bind and then it's going to be released as well. And so if you have an reversible inhibitor, excuse me, that binds to an active site, we say it's competitive. It's a competitive inhibitor, right? Because it's fighting for the same active site where the substrate would bind. And if it's non-competitive, well, that means it's binding at some kind of allosteric site, okay, where it's not the active site in the enzyme. All right, so let's take a look at example 12.9. It says chymotrypsin is an enzyme that helps digest proteins. The chymotrypsin inhibitor tosylphenylalanine chloromethyl ketone, TPCK, is shown below side by side with chymotrypsin's proper substrate. Given that the inhibitor permanently covalently bonds to a histidine residue in the enzyme's active site, TPCK can be classified as an irreversible inhibitor. So again, it's covalently bound, so it's irreversible. Further classify TPCK as a competitive or non-competitive inhibitor and provide your reasoning for your decision. So first of all, there's two major reasons why TPCK is going to be a competitive inhibitor. So I'm going to put here competitive inhibitor. The first clue is that the TPCK molecule has that phenyl group highlighted in the yellow box, which is identical to the natural substrate. And so that means the TPCK is going to mimic the natural substrate, right? They're kind of similar. The second and more definitive clue is that the TPCK is going to bind to chymotrypsin in the active site. And so if it's binding in the active site, that means it's competing with the substrate for binding at the active site, confirming again that it is a competitive inhibitor.